So, hello everybody, I'm Professor Brigitte Autran from Paris, and I'm glad to show you some data, encouraging or not, about HIV vaccine development and the challenges that are raised against this vaccine. So the first point is, why haven't we an HIV vaccine yet? And in fact, it's not a failure of researchers. It's not a lack of researchers. It's really a failure. And this failure is due to the fact there are many obstacles against HIV vaccines, and the main obstacle is the capacity of HIV to escape. There are several mechanisms for the virus to escape the immune system and globally the host control. And the first mechanism is the retroviral nature of this virus, which is a retrovirus, which is integrating his HIV provirus DNA into the host genome forever. And in the cell that it is infecting, it is behaving as a Trojan horse and liberating billions of HIV particles at any time. And among these billions of HIV particles, there is a very high number of HIV variants because there are some errors in the capacity of reproducing the quality of this HIV RNA because of lack of correction enzymes. And this is the reason why the HIV viability is so important, and it is particularly important in the epitopes, which are recognized by antibodies and by T cells. And finally, the third mechanism, it's a physical mechanism, combining also the question of the variants and leading to a weak capacity to protect of neutralizing antibodies. Altogether, these obstacles in the natural course of the disease are also preventing the cure of HIV, and this is a unique situation for developing a vaccine against a disease for which it is impossible to be cured. So there is no other example in the history of vaccine of an efficient vaccine against a disease from which we cannot recover. And it's raising the question of whether or not it's feasible to generate an HIV vaccine. But let's say it should be feasible, and let's see in more deeply what are the mechanisms of these obstacles. So as I said previously, the retrotranscription and the integration forever in an infected cell is a major obstacle, and it is constituting a reservoir of silently infected cells, which are remaining completely silent to any immune mechanism, to immune recognition, both silent to antibodies, silent to T cell recognition, because in addition of the lack of antigenic expression, there is a downregulation of HIV class 1 and class 2 molecules. So a kind of sanctuary in the infected cells. The other point is that this virus is penetrating the immune system at a high speed, which is not as frequent as that, and it's a major obstacle for vaccines. And you have here the example of the virus penetration through the vaginal mucosa. And you know, the vaginal mucosa has no lymphoid organs, but there are some macrophages and dendritic cells which are patrolling here. And those cells which are CD4 positive are binding the virus particle, at least on their cell surface. And by nature, these antigen-presenting cells are going to the draining lymph node, and there they present the virus to the CD4 T lymphocytes. So the CD4 T lymphocytes are infected within 1 hour. And as you know, the lymphoid organs is a completely homogeneous, even if it is sparsed, organ. So there is a continuous circulation within the different lymph nodes. And as soon as one lymph node is infected, you can consider the whole immune system is infected. And it's true that the systemic dissemination is occurring in less than 1 week. And during this first week, the immune system is mounting an immune response, but it's already too late, and it is still too little at this very early phase of immune recognition. The second obstacle is the variability, as I said. So there are two levels of variability. One are the clades. So clades are not a real issue for vaccine development. We have several clades vaccine against pneumococci. We have several clades vaccine against HPV, so it's not a big deal. It's simply more expensive. The big deal is here, which is the huge variability of the envelope at a molecular level. And you can see that on this scale, you have a comparison of the global variation of influenza over the planet during 1 year. And you see it is comparable to the variants which can be discovered in a single HIV individual at 6 years, for example, after infection, at a time where the virus is highly variable in a patient. So you see that this patient who is infected here has to change his immune system every day against the virus, as we have to change the influenza vaccine every year because of the variants. And if you consider the scale of variability of the envelope in a country during a year, you can see that it's absolutely overwhelming. So there is a single solution for a vaccine to be developed, is either to ignore the envelope, which is not possible because it is a target of protecting or supposedly protecting antibodies. So we have to find the conserved areas in this envelope to provide targets for the immune system and try to generate antibodies and protecting antibodies. And it's known from many vaccine models that the best antibodies in a preventative vaccine are neutralizing antibodies. In the case of HIV, we know that there is a very poor efficacy of at least the first generation of neutralizing antibodies that were detected several years ago. Why are these NABs so poorly efficacious? In fact, it's because the epitopes recognized by these neutralizing antibodies are hidden and are poorly visible to the immune system. So you have here the virus particle in gray and the spikes of the GP120 in orange and GP41 in pink. So you can see that the virus is approaching the cell membrane and binding to the CD4, and then binding to the CCR5, and then fusing the GP41 through the membrane. The point is that the CD4 binding site is highly conserved, so it's the ideal target for a vaccine. But the problem is that this CD4 binding site is completely hidden. And you have here a cut of the trimer of GP120, and this cut is open. But in the real life, the GP120 is highly flexible, it's moving constantly, and the CD4 binding site is always closed. And it is opening only when the virus is close to be binding to the CD4. So it's only at the last second that the CD4 binding site is opening and the immune system can see it. So there is a very short slot of time for the antibodies to penetrate to this CD4 binding site and block and neutralize the entry of the virus. Nevertheless, it has been shown that other epitopes than the CD4 binding sites might be also good epitopes for the neutralizing antibodies. And particularly, it has been shown that glycans on the surface of the glycoprotein, even if they are extremely poor uh, epitopes usually, some of these glycans can be recognized by highly efficient and broadly neutralizing antibodies, but they are very rare, and you have to remind that um, most of this glycan is composing a kind of shield. We speak of a glycan shield, which is protecting the envelope from the action of antibodies. But happily, the GP41, which is in pink here, is less variable than the GP120. And we have several epitopes on the GP41 region close to the membrane that can be targets for neutralizing antibodies. So 20 years ago, there was a discovery of 4 human monoclonal antibodies from patients. And these monoclonal antibodies were mapped and were shown to be directed either against the CD4 binding site or against a glycan epitope on the surface of the GP or against 2 epitopes of the GP41. These are the generic BNAbs, broadly neutralizing antibodies, that compose the first generation of antibodies and that give some hope that perhaps it might be feasible to generate a vaccine based on neutralizing antibodies. Because indeed, as I said, if you want to be preventative, you have to have neutralizing. The best tool are the neutralizing antibodies, which are the only tools capable to block penetration of the virus. Otherwise, you can have also, of course, CD8 T cells and, of course, CD4 T cells, which are always necessary to generate BNAbs and CD8 T cells. But if you have a vaccine generating CD8 T cells alone, these CD8 T cells will recognize the virus antigens only once the virus has infected cells. So only once the virus has integrated its genome in the host genome. So it's a bit too late. And the best you can hope with a CD8 T cell-based vaccine is to lower the level of replication and the residual level of replication after the primary infection, and then to delay the appearance of the disease, which, as you know, in the case of HIV, is a lethal disease due to AIDS. So I will not speak about therapeutic vaccines because for the sake of time, it's a bit too short. So to generate a vaccine, we like to have, and we need now, in a rational design of vaccines, we need to have immune correlates of protection generated from observations of the natural course of the disease. Regarding antibodies against HIV, as I said, it has been extremely difficult to demonstrate until recently the occurrence of neutralizing antibodies, but it was possible. And nevertheless, there's still no significant relationship between neutralizing antibody levels and virus control in already infected individuals. But however, if you perform some passive transfer of neutralizing antibodies, either in humans or in macaques with a Hiv chimeric model of infection, you can show that neutralizing antibodies, monoclonal antibodies, are capable of controlling, at least partially, the virus. It has been shown 20 years ago with the first generation of antibodies. It has been shown more recently with the second generation of antibodies, and I will come back on that. But once, for the last 20 years, it was considered that the neutralizing antibodies would not be easy to generate, and that CD8 T cells might be a better immune correlate of protection. And there was some demonstration that indeed there were interesting targets for vaccine. And indeed, when you deplete macaques from CD8 T cells, you lose the capacity to control the virus, suggesting that CD8 T cells are really playing a role to control the virus. In humans, of course, we cannot deplete the CD8 T cells, but it was shown that there was good correlations between either the magnitude of the CD8 T cells against HIV at very particular steps of the disease, particularly during acute infection, and also in a group of individuals who are called the elite controllers or long-term non-progressors, who are not progressing towards AIDS because of their strong CD8-based immunity. It was shown also that GAG-specific CD8 T cells were more potent than envelope-specific CD8 T cells and directed against more conserved epitopes. And it was shown that the quality of the CD8 T cells in terms of memory was also a good parameter, and of course the affinity of the CD8 T cell receptor for the antigens. And as for any infection, the CD4 T cells are obviously extremely helpful and necessary to generate good CD8 T cells and good antibodies, even if they are not protective per se. So in terms of vaccine development, what has been tested? Everything, absolutely all approaches, all classical approaches of developing a vaccine have been tested. I have no time to go into the details. All failed. And this was the reason why in the mid-90s, it was proposed to use an intermediate strategy, which is now very, very common, the strategy of using a replication deficient viral vector, so exactly the same that we are using for SARS-CoV-2, but it was a kind of revolution at that time. And there were two families of vectors that were prioritized at that time, either the pox virus family derived from the vaccinia construct, but less pathogenic than the pox virus, and the adenovirus family, as now. And there were two strategies that were proposed. The first strategies that was built at a time where it was considered that there was no possibility to generate antibodies and neutralizing antibodies with a vaccine. And the concept of a pure T-cell based vaccine was prioritized and probably easier to generate. So the strategy was composed of using a recombinant vector alone or combined to DNA bearing coding for the same antigens to activate primarily the T cells. And soon after, or even at the same time, was proposed a prime boost strategy where a recombinant vector was associated to a kind of boost with the soluble envelope in order to boost the B cells at much higher levels and to produce both CD8 T cells and CD4s and B cells and high levels of antibodies. So to make a long story short, the first strategy was using an adeno5 mostly, not only, but mostly. And this adeno5 construct in the first big trial of HIV vaccine, as you see here, was recombined only for GAG, POL, and NEF genes, so no envelope. So it was a purely T-cell based vaccine with no goal of generating antibodies. You have here the design of the phase 2/3 trial that was generated. It was a step study. And this trial had to consider whether or not the volunteers were pre-exposed to adeno5 and had adeno5. Again, to be very short, unfortunately, this trial, the STEP study, had to be stopped at half the trial, definitely stopped, because of an increased frequency of HIV infection in the vaccinees compared to the placebo. These differences were not significant, but at least there was futility, if not failure, and the trial was definitively stopped. And similar levels were observed in a similar trial using the same construct in South Africa with exactly the same results. And several years later, there was another trial that was testing adeno5 recombined with the same genes plus the envelope, plus a DNA prime, in order to boost even more deeply the T cells. And this trial was also prematurely halted, also because of futility and with the same trend of higher frequency of infection in the vaccinase compared to the placebo. And there was no way to analyze any factor, any immune factor that could explain clearly this failure, nor give some hints toward some apparent control. Happily enough, several years later came out the results of another big trial, 16,000 individuals large trial in Thailand, using not the adenoconstruct, but a poxvirus construct, the ALVAC, derived from the canary pox vaccine. It was a prime boost strategy incorporating the envelope gene in addition to the GAG, the pollen F gene, and a boost with the soluble protein. And for the first time in HIV vaccine history, there was some success in this trial with 31% vaccine efficacy. It's not huge at 3 years. It was significant, but you can see that it was observed in a very limited number of individuals. And the conclusion that this vaccine may reduce the risk of infection in a population with heterosexual risk, but there was no effect on the virus load or on CD4 count after HIV infection. So no protection against the disease in the individuals who were vaccinated but infected. Finally, some correlates of protection could be proposed in this trial, showing that the envelope-specific antibodies were associated to this low level of control. It was not mediated by neutralizing antibodies, but by non-neutralizing antibodies that were directed against the V2, the variable, one of the variable region of the envelope. From this study, from these results, which were extremely important in the development of HIV vaccine, came out a new strategy showing that it was feasible to generate antibodies and to generate some protection linked to antibodies. And there were two routes, either to generate neutralizing antibodies or to generate functional antibodies, but non-neutralizing antibodies capable to mediate other functions than neutralization. Happily enough, because of other breakthroughs in the capacity to generate and to isolate monoclonal antibodies from human cells, from patients, it was possible to discover a much broader range of broadly neutralizing antibodies capable to neutralize large numbers, if not all, clades of HIV and all subspecies of HIV. And these broadly neutralizing antibodies, interestingly enough, were directed mostly against exactly the same epitopes than the ones that had been described 10 or 20 years before: the CD4 binding site, a glycan epitope, and two epitopes on the GP41. So the mapping of epitope that had been generated 20 years ago was still true. But the advantage of this new generation of BNAbs was that they were active at much higher levels or at much lower concentrations and had a far broader spectrum of action than the first generation of neutralizing antibodies. But these antibodies, even if they are frequently observed in HIV-infected individuals, they are extremely rare if you consider the truly broadly neutralizing antibodies that are capable to neutralize all the three tiers of different HIV particles. And these broadly elite neutralizers are observed in only 1% at the most of HIV-infected individuals. And even when these BNAbs are detected in HIV-infected individuals, they are always appearing late in the disease, usually during the second year of infection. And when they are isolated in viremic individuals, and they are not correlated to disease protection, suggesting that they are not efficient enough to protect the patient against his own viruses. And in addition to that, the genetic analysis of the sequence of these antibodies showed that these antibodies had a very long CDR3 region, which was associated with a lot of somatic hypermutations, suggesting that it was very peculiar antibodies that had this capacity to be broadly neutralizing. And to reach this capacity of broadly neutralizer, these antibodies needed an accumulation of somatic hypermutations in their genes, explaining why it takes so long time to generate these antibodies, and suggesting that the immune system has to be educated against different variants before being capable to generate broadly neutralizing antibodies. So in terms of vaccine development, you can understand it's a concern. But the first step after generating these BNAbs was to demonstrate they could be efficacious. And indeed, when they were infused as a monotherapy in HIV-infected individuals, you can see that these antibodies were efficient to decrease the virus load by 1 or 2 logs, but in fact, they were transiently efficient, and not only because of the short half-life of antibodies, but also and mostly because of variant escape. So if you inject a single BNAb, it will unfortunately expose, even if it is broadly neutralizing, it will expose to immune escape by other variants in almost all cases, suggesting that, of course, a vaccine should not target a single epitope, which is absolutely obvious. So we are now in the rational design of HIV vaccine. So to generate neutralizing antibodies against the virus, you have to target these 4 major epitopes. There are other sites recognized by BNAbs, but these 4 targets, 2 on the GP120 and 2 on the GP41, are extremely important. So, 3 on the GP120, with the majority of antibodies directed against the CD4 binding site, and 2 epitopes in the GP41, which are described here. So now we know what are the epitopes, but still we cannot develop an HIV vaccine capable to generate BNAbs. Why? In fact, all attempts so far at developing BNAbs using either adjuvanted envelope proteins or vectored envelope, followed by adjuvanted envelope protein in a prime boost strategy, failed to generate BNAbs. Why is that so? Probably, even if the envelope is engineered, the presentation of critical epitopes on the immunogen is not absolutely comparable to what is observed in the native envelope. Second, you have the problem of facing immunodominance of highly variable surface regions compared to the conserved regions. So the immune system will recognize primarily the highly variable regions, and it will take time to generate antibodies against the conserved region. And in addition, for reasons that are not well understood, it appears that in the natural human repertoire of antibodies capable of recognizing HIV, there are very low numbers of high affinity germline antibodies capable to give rise to broadly neutralizing antibodies. So we need to have high selection mechanism to provide high selection mechanisms with the vaccine schedule of immunization to be capable to generate these BNAbs. So it's not only a question of immunogen, it's also a question of how you will immunize the immune system once you have the good immunogen. And this is illustrated here. You have different proposals of building an HIV immunogen, HIV envelope immunogen, to expose ideally either the CD4 binding sites or other epitopes of the envelope. And so far, it's not yet clear what is the best component that would be elected for an HIV vaccine. And most of the best components are so far capable to generate tier 2 neutralizing antibodies, but no tier 3 neutralizing antibodies, which means that these antibodies would be capable to recognize 2/3 of the HIV particles, but not all of them. So you have another cartography of these different attempts of producing immunogens. You have here the results of one of the best immunogen that has been produced so far, which is called SOZIP, which is a stabilized envelope exposing the CD4 binding site, which is showing in macaques good results with tier 2 BNAbs, and it's currently in clinical trials in humans. But to generate the appropriate antibodies and the BNAbs, we have to accelerate the selection of high affinity antibodies and to expose the immune system to a different variance in the time course of the primary immunization. So the idea is to use successive immunization with different variants of the same immunogen in order to increase the number of somatic hypermutations in the immunoglobulin genes and to generate appropriate BNAbs. So this strategy is obviously highly expensive, extremely difficult to implement in humans, but nevertheless, it is currently being tested in clinical trials. And finally, as it is extremely difficult to generate neutralizing antibodies, it became more and more obvious that, well, okay, it's good to have neutralizing antibodies, but let's try other function of antibodies, and particularly antibodies capable to generate cellular cytotoxicity or complement-mediated phagocytosis. This is an area which is opening more hopes of success because it seems to be easier. And you can reach that either with envelope-specific antibodies, of course, but also you can imagine that some P24 antibodies might play a role. And you might hope that, okay, you might not be capable to prevent completely the infection if you don't have neutralizing antibodies, but you might be capable to decrease the level of replication and decrease the final infection of the patient. And this has been shown, reasonably been shown in macaque studies using a prime boost strategy with a recombinant adenovirus vector and a soluble GP140 molecule in macaques. And it has been tested also in humans, and it appears that the level of immunization of antibodies generated in humans is close to the one observed in macaques, and that the success, the preventative success of this combination of adenovirus vector plus GP120, 140 in preventing HIV infection might be reached in humans, but the clinical trials are ongoing and we don't have the results yet. So in terms of clinical development, everything is tested in terms of antibody-based immunogens. You have different trimers, you have germline targeting strategies, you have lineage-based strategies and epitope-based strategies. All are being tested so far in antibody-based vaccine approach that are either trying to target or to induce broadly neutralizing antibodies or to generate non-neutralizing antibodies. But it is considered that the difficulty of reaching the goal of preventing HIV or controlling HIV will be too difficult with a vaccine inducing only antibodies, and that a combination of antibody-based plus T-cell-based vaccine is mostly favored. And this T-cell-based vaccine will have not only to induce CD4 T cells, which are always mandatory, but also to generate CD8 T cells. And this is usually planned to be obtained by using vectors, either non-replicating virus vectors, mostly adenovirus or pneumopox viruses, or even live replicating vectors, such as ones provided with the CMV vector. And in any case, these vaccine candidates have to focus on a mosaic of conserved HIV antigens in order to generate a broader immunity and a stable immunity capable to recognize conserved epitopes and therefore more broader targets of virus. So if you consider the extremely rapid success of SARS-CoV-2 vaccine development compared to the failures of HIV vaccine, you say, what can we learn from SARS-CoV-2 vaccine development? And of course, the major lesson is provided by mRNA technology, even if it's certainly much easier to develop a vaccine against SARS-CoV-2 than against HIV. Nevertheless, we have to consider whether or not the mRNA vaccine technology might be useful. And indeed, the versatility of this mRNA vaccine technology might be extremely helpful in this necessity of presenting successive variants to the immune system in order to generate broadly neutralizing antibodies. And there, the mRNA vaccine technology is certainly the best technology to achieve this goal. And it has been shown in macaques that indeed the usage of lipid nanoparticle encapsulating nucleoside-modified mRNA vaccine coding for HIV might, and particularly GP160, combined to or compared to this SOSIP immunogen that I mentioned before, this mRNA technology might be more powerful to generate high levels of antibodies and long-term levels of antibodies. So certainly the technology of mRNA vaccine will help in the future development of HIV vaccine. So in conclusion, we can conclude that we are in a difficult position compared to other vaccine development. Certainly, we have to consider mostly the prime boost strategies, inducing neutralizing and or non-neutralizing antibodies combined to T cells, and the mRNA technology will change the game. I will not speak a lot about, or will not speak about therapeutic vaccine, which are depending on the same obstacles. And I would like to conclude with other challenges that are opposed to HIV vaccine. which is, even if we have a good immunogen, how will we evaluate the vaccine efficacy, considering the huge advantage of provided by treatment, not only treatment of HIV-infected individuals, but also prevention by treatment, what we call the PrEP, pre-exposure prophylaxis, which has been shown to have 90 to 95% protective efficacy. Even if it is not achievable everywhere in the world, or accessible everywhere in the world, the PrEP is, in a country like France, accessible to anybody. So how will you ethically accept to perform a vaccine trial against placebo in a country where PrEP is available? It's really a challenge. There are other mechanisms of prevention, such as circumcision, which can provide up to 50% protective efficacy against HIV. So to conclude, we are not at the end of the game. Not only it is still difficult to develop an HIV vaccine, an HIV immunogen, but it will be extremely difficult to test the clinical efficacy of this HIV vaccine. So I'd like to stop here. I'm still hoping that I am convincing you that it's still worthy to work on HIV vaccine, and that one day it will be possible to have an HIV vaccine. Thank you very much for your attention.